Hello student, I am Dr. Manish Kumar Gupta, working as an Assistant Associate Professor, Department of Biotechnology, Veer Bahadur Singh, Purvanchal University, Jaunpur, Uttar Pradesh. Today we will discuss about sequence alignment, specifically biological sequence alignment. Thus, in this talk, whatever the content I will discuss, the outcome will benefit to our students. They will understand the basics of computational challenges in biology, use of sequence alignment, notations used in sequence alignment, fundamental of scoring system of alignment, and the various types of alignments. Before going to the understanding of the sequence alignments, one has to understand the basic fundamentals of biological system, specifically central dogma of molecular biology. Dear student, we know that our bodies contain cell, the cell contains nucleus, and nucleus contains a lot of informations in the form of a coded one, and the one coded is a DNA coded sequence, and these DNAs are composed of 4 different types of letters: adenine, guanine, cytosine, thymine, and once these DNA in the form of genes goes to the second information levels through transcription methods, they code another letter called AGCU, and this represents the messenger RNA. The information flows from messenger RNA to the next level through the process called translations. So, these translations lead to the formation of 3-dimensional biologically active molecules called proteins. These proteins can be made through a genetic code which are represented in the form of codons. So, there are total 64 codons, out of which 3 ones are stop codons or termination codons. So, this central dogma tells us about the basic flow of information from DNA to protein in the form of amino acids. But before going to the next level, one has to understand the differences between the expressions of genes between the prokaryotic system and eukaryotic system. So, as we know that in prokaryotic gene expressions, the prokaryotic genes lead to the formations of mRNAs which do not contain any kind of a exons or introns, and they lead to the formations of proteins, and these proteins form through the co-translation mechanisms, whereas in case of eukaryotic gene expression, the gene lead to the formation of heterogeneous nuclear RNA in the nucleus process, and in nucleus through the processing and splicing it lead to the formation of mRNA in cytosol. So, this mRNA lead to the formation of protein which are in the primary structures, the linear one, then secondary structure, the tertiary structure, and these tertiary structure lead to the biologically active molecules proteins. So, this is the mechanism of the gene expressions functions T, because only G A is contiguous and T is after the G. Therefore, it cannot be considered as a substrings or subsequence. The other notation is prefix, which is a substrings containing the first character of a string containing the empty string. For example, in the string GCU, a prefix may be the empty string G, GC or GCU. The next notation is a suffix, a substring containing the last character of a string. For example, G is a suffix of UUG. So, coming to the next point, the alignment scores similarity, identity versus distances. So, what is the similarity in the sequence alignment? It is both quantitative and qualitative aspects. Similarity is quantitative in nature, saying that two sequences show a certain degree of similarity, whereas an alignment is a mutual arrangement of two sequences, which is a sort of qualitative answer that exhibit where two sequences are similar and where they differ. A similarity measure is a function that associate a numeric value with a pair of sequences. For example, higher value is equal to higher similarities. So, score can be a positive or negative value, and optimal alignment maximizes score. Similarity is also equal to resemblance between two sequences of characters when compared. An optimal alignment maximizes similarity, and it implies that large values are good. The next point is a identity, the extent to which two nucleotides or amino acid sequences have the same residues at the same positions. A number of characters that match exactly in an alignment. is called identity. So, coming to the again the distance point, the third one. So, the distance measure is a function that associates a numeric and non-negative value. The optimal alignment minimizes the distance, larger the distance value, smaller the similarity. So, the distance can be measured by two methods, one is called Hamming distance, the other one is called a Levenshtein. So, the Hamming distance is the two sequences of equal length, the number of characters in which they differ. This Hamming distance method is not flexible and has no fixed correspondence between their character positions. For example, let us say the two sequences, the first one is the source sequences which is represented as a S, the target sequences T. So, let us count how many characters they differ. For example, in the source sequences A, G, G, A, the G G is represented in the red color, and in the target sequence it is A, T and T. Here one must understand the length of the two sequences should be same. So, at the second position where A varies from the G in the source sequence, T vary from the source sequence. So, there are two difference in the character. So, the Hamming distance between the source and target is 2. In the second case, the length of the characters is 5 and there is a differentiate from the CGT which is indicated in red color. In the target sequence it is a GTA. The first position which is a T and the fifth position which is a T, they are same. So, the Hamming distance in which the characters numbers differs is a 3. The third case is where the length of source sequence is a 9 and the length of target sequence is 9, but only 2 characters are matching and 7 characters they got differ. So, the differences from the 7 characters is nothing but the Hamming distance between the source and target. So, performing this Hamming distance, one has to also understand about the edit operations. What is the edit operations? The minimum number of operations required to get the optimal alignment score between two sequences is called the edit operation. So, the edit operation by introducing a gap character, match, mismatch and substitution in a given two sequences of source and target, and say that the pairs of characters. For examples, in this example, let us say A and A denotes match, no change in the character in source and targets. When I say source and target, suppose you are having a sequence of source and sequence of targets. So, at particular positions there is a, let us say, guanine in the first sequence and guanine in the second sequence at position number 3. So, this will be treated as a match. So, the second case is a comma gap which denotes the deletion of the character in target sequences. Here the A which is indicating at the first position is treated as a character found in the source and the second position is represented in the target. So, therefore, the gap will represent as a deletion in the second sequences or target sequences. The third case is where the two character in the two sequences do not match. This will represent as a substitutions of characters. So, in the source the character is A, in the target the character is B, where A and B are not equal. So, they will treat it as a substitution, or sometimes it is also called as a replacement. The fourth case is the gap is aligned or matched with the another case which denotes the insertion of character in the target sequence. So, they can be represented by the two examples where the edit operation performs. For examples, in the first case you can see the source and target which are indicated in the red colors. The source is having a character A, G, C, A, C, A, C and gap and A. So, this is a source which is a character of, you can count the character from first position A, second position G, third position C, fourth A, fifth C, sixth A, seventh C, 8 is a gap character and 9th is a character A. So, the length of character in the source is a 9th. Similarly, the length of character in the target is 9, but in target at position number 2 is there is a gap. But taking the source as a reference sequences, the second position will be treated as a deletion of the characters which is indicated as a D, and at position number 8 you can see there is a insertion of character T in the target sequences. So, that will be treated as a insertion with reference to the source sequence. The M is representing the match character, the red color indicated D is deletion, and the I represent the insertion of the character. So, there are two edit operations required in this example. Similarly, in the another case where you can see the length of character, once again you can count from the source and target. The source is A, G, gap, C, A, C, A, C, A. So, the length of character is 1, 2, 3, 4, 5, 6, 7, 8, 9. Again the length of character is 9, but in the source at third position there is a insertion corresponding in the target sequences, and similarly at position number 8 there is a deletion of the character, and at position number 2 there is a substitution case, and at position number 1, 2, 3, 4, 5, 6, 7. At position number 7 the A is substituted with T. So, there are two substitutions, 1 insertion, 1 deletion, 5 match are there in the second example. So, this is all about the edit operations. So, the second model is a Levenshtein distance model, or sometimes it is called a unit cost model. So, the edit protocol is to measure distance by assuming a cost or weight W to each operation, where W A is equal to 0 for match character and W A and B is equal to 1 for when the character A is not equal to character B, and this can happen is a mismatch, substitution or replacement case, where the weight or cost W, the gap character aligned with the character, or weight character aligned with the gap is equal to 1 in the case of insertion or deletion. In short, it is also known as a indel operation. So, the cost of alignment of two sequences, source and target, is the sum of the cost of all the edit operation that lead from S to T. The cost should be minimized and have positive values. One has to understand it should be minimized and have a positive value. And optimal alignment of source and target is an alignment which has minimal cost among all possible alignments. The edit distance of S and T is the cost of an optimal alignment of S and T under a cost function W. It is denoted as DWST. So, once again you can see there are two example has been taken. So, the source and target. So, at the second position there is a deletion, and at the 8th position there is a insertion. So, the cost will be 2. Similarly, in the second example the length is same, but there is a 2 substitutions, 1 insertion and 1 deletion. So, the cost will be 4. So, the edit distance, but the edit distance is equal to 2 in the first case. So, coming to the further process, what are the different types of methods involved for the sequence alignment? So, the dot matrix analysis, dynamic programming algorithms, and heuristic methods includes BLAST and FASTA. So, the dot matrix analysis is it displays any possible in sequence alignments as diagonals on the matrix. Best way to see all the structures in the commons between the two sequences, and the dot matrix also used to view repeated structures or inverted repeats in a single sequences. So, you can see in this diagram wherever there is a match in the character, a dot will be put it in the matrix, and on the diagonal sets if it is a continuous matching you can draw a line, you can see if it is a continuous matching you will see there is a continuous diagonal of dot, and such matrix is called a dot matrix. The second method is a dynamic programming algorithms. So, this algorithm is a central to the computational sequence analysis. The dynamic algorithm efficient recursive method to search through all the possible alignments and find the one with an optimal score. Dynamic programming algorithm also usually consists of a recursive relation, tabular computations and traceback. As you can see in the table, there is a two sequences, source and target sequences, and there is a matrix, and the matrix first line and the first row and the first column is called a 0th row and 0th column. So, there is a some values you can see, and at the last cell there is a value from which is the alignment score. The heuristic methods includes BLAST and FAST-A. The BLAST and FAST-A heuristic methods that attempt to approximate the optimal local similarity shared by two sequences. The goal of these methods is to search as small a fraction as possible of the cells in the dynamic programming matrix, while still looking at all the high scoring alignments. So, what are the alignment types? The alignment types can be categorized in a sequence alignment and a structure alignments. Structure alignment, which is only used for known sequences whose structures are available. They use the method called distance matrix alignments, DALI, sequential structure alignment program, SSAP. So, this can be further classified. One can see alignment can be categorized in a structural alignments and sequence alignment, and the sequence alignment can be further categorized sequence genome, genome alignment, and the sequence genome can be further classified short reads and long reads. And this sequence alignment further, when we are taking a two sequence which is a homology detection and the multiple sequence alignment, and another is a optimal alignment dynamic programming and pair hidden Markov model. So, the classification of alignment problems, the hierarchy classifies various forms of alignment problems. Our proposed approaches belong to the yellow marked subgraph of the hierarchy. So, again coming to the taking only the sequence alignment, which can be classified in a pairwise sequence alignment and multiple sequence alignment. The pairwise sequence alignment further classified in the global alignment, local alignment and free space alignment, whereas multiple sequence alignment can be classified also global alignment as well as local alignment. So, the multiple sequence alignment methods include the dynamic programming, progressive methods, iterative methods, motifs or profile methods. What are the uses of pairwise and multiple sequence alignments? The pairwise useful for finding the conserved regions between the two sequences. L-align, BLAST, emboss needle, emboss water are the example of pairwise sequence alignment biological tools. Multiple sequence alignment is a useful for finding regions of variability or conservation in a family of proteins, phylogenetic analysis, detection of a homology between newly sequenced genes and an existing gene family, and prediction of protein structure. The examples of multiple sequence alignment tools are muscles, T-Coffee, MAFET, Clustal W, Clustal Omega. So, one has to also understand when we are going for a sequence alignment, the basic fundamentals of gap score or gap penalty. What is a gap? A gap is any maximal consecutive run of spaces in a single sequence alignment. Gap helps to create alignments that better conform to underlying biological models and more closely fit patterns that one expects to find in meaningful alignments. The length of a gap is the number of indel operations in it. The number of gaps in an alignment is denoted by number gaps. For example, the two sequences which are represented in a S and T, the S composed of sequences A, T, T, C, then one line, again line. This hyphen line is nothing but a gap together, then G and A, then hyphen, then T, G, G, A, C, C. Then in the target A hyphen hyphen C, G, T, G, A, T, T hyphen 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 C, C. This hyphen is nothing but the total you count as a one gap. So, there are 4 gaps and the consecutive run of this is called spaces. So, if you count the total number of hyphen in the first sequence source, there is a 3 space, and in the second total is a 5. So, total spaces in this 2 source and target is a 8 spaces, 4 gaps and 0 mismatch, 7 match. So, how we can deal with this gap in the sequence alignment? We have to adopt gap penalty models. So, there is a called constant gap penalty models. Each individual space is free. Constant weight W g for each gap independent of its length is given to an alignment that maximizes this source and target sequences plus the gap number of gaps multiplied by the constant weight. The other model is called a affine gap penalty model, where a gap is given in 2 weights. One, W g is the weight to open the gap, The other one W s is the weight of extend the gap or extension of the gap with one more spaces. So, the total penalty for a gap of length q is W total is equal to W g plus q W s. After the above affine formula, the model is called affine gap penalty model. So, the alignment in which it maximizes the summation of total number of characters source and target plus the weight to the open of the gap multiplied by the number of gap plus the weight of the extension of the gap, that is the W s multiplied by the number of spaces. So, as you can see in this structure that in sequence 1 and sequence 2, that if you count the distance between these two sequences and keeping the value match 0, substitution 1 and insertion and deletion value 2, you will find the distance, total distance is equal to 14, in which the total 4, because there is no match value is given. So, we are giving a total 4 multiplied by 1 substitutions plus the indel value 2 multiplied by 5, because there are a total 5 indel operations are there. So, you can see that in sequence 2 there is a 4 gap and in sequence 1 there is a 1. So, total is a 5 indel values. Similarly, if you take the other example distance 2, if you give the match for adenine and thymine and guanine and cytosine is equal to 1 and adenine and guanine is equal to 1.5 and indel is 2. So, the distance will be 3 plus 1.5 plus 2 into 5 gap. Similarity can be identified given a match value 1, substitution 0 and indel 1.5 will be giving a 24 matches are there plus minus 1.5 indel value multiplied by 5 number of gaps. So, the score will be a 16.5. So, the similarity score is a 16.5, where is the distance 1 is a 14 and distance 2 is a 14.5. So, the general setup is a substitution matrix S I j indel S I minus I hyphen or S gap j. So, this is all the basic about the sequence alignments. So, students, thank you very much for listening this talk. You can go through a websites on SWAM and you can see all these contents are available there. Thank you.